So good evening everyone. I'll start with the session. So today we'll be going on week 6 live session on Bioinformatics Algorithms and Applications course. So in this session I'll also cover a demo on CD hit which I missed in the last class. So initially, to start with the protein structure. Protein structure is generally divided into four levels, primary, secondary, tertiary and quaternary. Of these four levels, tertiary and quaternary structures refer to the 3D and higher order structures of the proteins. When we particularly look into the tertiary structure of a protein, it refers to the overall 3D shape of a single polypeptide chain. So this tertiary structure is determined by the interactions between the side chain of amino acids that make up the protein. So these side chains can interact with a variety of chemical bonds and forces, including hydrogen bonds, hydrophobic interactions, electrostatic interactions, and disulfide bonds. So these are termed as non-covalent interactions. So these interactions cause the protein to fold into a specific shape, which is stabilized by interactions between the amino acids. This tertiary structure of a protein is crucial for its function, and the shape of the protein determines how it interacts with the other molecules. When we come to the quaternary structure of a protein, it refers to the arrangement of multiple polypeptide chains that make up a functional protein. These subunits can be identical or different and are held together by various interactions, including hydrogen bonds. The quaternary structures is important for function of many proteins, particularly enzymes and signaling. So for example, here I am showing a hemoglobin, which is responsible for carrying oxygen in the blood and consists of 4 subunits that are arranged in a specific way to allow oxygen binding and release. In summary, the tertiary structure refers to the 3D shape of single polypeptide chain, while the quaternary structure refers to the arrangement of multiple polypeptide chains that make up a functional protein. Both the structures are important for the function of the proteins and can be affected by changes in their amino acid sequence and environmental factors. So here I have some questions. So the first one starts with the which level of protein structure involves the linear sequence of amino acids? Which level of protein structure involves the... Thank you for your answers. The primary structure of a protein refers to the linear sequence of amino acids joined by peptide bonds. Next, the alpha helix and beta sheets are example of which level of protein structures? As we have checked the secondary structure prediction methods in the last class, the alpha helix and beta sheets are the example of secondary structure of the proteins. So these secondary structures refers to the local folding of the polypeptide chain into regular structures like alpha helices and beta sheets. Most of these secondary structures are stabilized by hydrogen bonds. Okay. So hemoglobin, which we discussed example in our first slide. So hemoglobin composed of 4 subunits is an example of which level of protein structure? Hemoglobin composed of 4 subunits is an example of which level of protein structure? Those who have joined late, we have started with the explanation to protein tertiary and quaternary structure. So hemoglobin is composed of 4 subunits is quaternary structure because quaternary structure refers to the arrangement of multiple polypeptide subunits in a protein complex. So as hemoglobin consists of 4 subunits, each with its own tertiary structure, and their arrangement constitutes the quaternary structure. So how these structures have been derived? So what experiments? So little understanding about the experiments is required in bioinformatics as well. So firstly, we'll start with the X-ray crystallography, which is widely used method. So X-ray crystallography is like taking a detailed photograph of a protein structures. So proteins are molecules that are made up of atoms. So when X-ray passes through a crystal made of a protein, they diffract. That is, they bounce off in patterns that depend on the arrangement of atoms in a crystal. So this diffraction pattern refers to the arrangement of atoms in a crystal. So this technique of X-ray crystallography provides high resolution images of protein structures at the atomic level. It is often used when researchers need precise details of how atoms are positioned within a protein. Next, we have NMR. So NMR stands for nuclear magnetic resonance spectroscopy. So NMR spectroscopy is similar to listening the subtle variance of vibrations and variations of atoms within a protein. When a protein is placed in a strong magnetic field or a radio waves, the atomic nuclei within the proteins resonate at a different frequencies based on their environment, revealing details about the protein structure. So this NMR techniques provides information on interactions between the atoms in a protein and its surrounding environment. It is particularly useful for studying proteins in solution, offering insights into dynamic properties. Next, we have electron microscopy, so which involves bombarding the proteins with a beam of electrons. So these electrons interact with the protein, generating an image that reveals its structural features. Then we have CD spectroscopy, that is circular dichroism spectroscopy. This mostly focuses on assessing the secondary structure of a proteins, focusing on their helical and sheet-like arrangements. So this CD spectroscopy works by measuring how differently polarized light is absorbed by the protein. Next, we have fluorescence. So this technique involves tagging proteins with fluorescent molecules that emit light when excited by a specific wavelength. So by measuring the emitted light, researchers can gather the information about position, tertiary structure, and dynamics. So I think I have some more questions here. So which method involves bombarding protein crystals with X-rays to analyze their diffraction pattern? So understanding about these experimental patterns and their role in generating the 3D structure of an image is crucial for developing some feature calculations or structure-based feature calculations in bioinformatics. So why these basics are necessary? Okay, yeah, it is X-ray crystallography, which involves bombarding protein crystals with X-rays and analyzing the diffraction patterns generated. So wherein these diffraction patterns provide information about the arrangement of atoms within the crystal, which allows one to determine the protein structure. So then we have which technique utilizes the behavior of atomic nuclei in magnetic field to determine its protein structure. So the more information on these techniques will be available in the structural biology course of NPTEL by Saugata Hasra. So for more details of protein structure prediction experimental techniques, you can refer those. Even I will be referring those. So it is NMR spectroscopy. So NMR spectroscopy utilizes the behavior of atomic nuclei and magnetic field to determine the protein structure. So which technique focuses on differential absorption of left and right circularly polarized light to determine protein secondary structure? Yes. That is CD spectroscopy, which focuses on differential absorption of left and right circularly polarized light, so which provides more information about protein secondary structure. Then I think I have one more. No. Okay. So more detail, we'll be studying about X-ray crystallography. So when we observe till now, what are the structures generated? We see most of the structures are generated using X-ray method. Now currently cryo-EM is also the number of structures determined using cryo-EM is also increasing. But till date, X-ray crystallography based determined structures are many in the databases. So the steps involved is protein purification, crystallization, data collection, data processing, model refinement and validation. The first step is to obtain the pure sample of the protein, so which is done by typically expressing in any host organisms, and then purified using a combination of chromatography, filtration and dialysis methods. Then the protein must be crystallized to form regular and ordered array of molecules that can be analyzed using X-rays. So here the concept of crystallization comes. Here crystallization is a difficult and time-consuming process that is often requires screening many different conditions such as pH, temperatures or concentration to find the optimal conditions for crystal growth. Once crystals have been obtained, they are mounted onto a goniometer and placed in a beam of X-rays. So these X-rays are diffracted by the atoms in the crystal, producing a diffraction pattern on a detector. So the goniometer is rotated and diffraction data is collected at many different angles to generate a complete data set. Finally, we have data processing. So the diffraction data that we have obtained must be processed to extract the structure of a protein. This again involves several sub-steps, including scaling and merging the data from multiple crystals, calculating the phases of diffraction, and building an initial node. Then we have model refinement and validation to confirm with the 3D structure of the protein. Okay, now that several structures may be solved using this experimental technique, but where to store those, how to access those? We have PDB. PDB stands for Protein Data Bank. So Protein Data Bank is a database that contains information about three-dimensional structure of proteins and nucleic acids and other biomolecules as well. So we term PDB as a key resource in the field of structural biology, biochemistry and biophysics, providing access to experimentally determined structures of the proteins. So this PDB is maintained by Worldwide Protein Data Bank organization, so which is collaboration between several research institutes such as RCSB, that is Research Collaboratory for Structural Bioinformatics, Protein Data Bank in Europe, PDBE, and PDBJ, which is Protein Data Bank of Japan. So the file format used by PDB. So why should we have a unique file format? So this is to generalize the protein structure concept. So why there's a particular format for a PDB file. So the format of a PDB file is divided into several sections, including header section, which contains a brief description of the structure, atom section, which contains information about atomic coordinates and properties of each atom, and the connect section, which contains information about the connectivity between atoms in the structure. So each section will be dealt. So the first is the atom section. So atom section will have atom serial number, atom name, residue name and chain identifier, residue sequence number and XYZ coordinates of the atom, occupancy and temperature factor. So then we have connect section of PDB, which contains information about bonds between atoms in the structure as well as other types of connectivity. So to summarize about PDB, PDB is a database of experimentally determined protein and biomolecule structures, and PDB file format is a plain text that contains information about atomic coordinates, properties of each atom in the structure. So the PDB file format is, we'll see visualize how PDB looks. So let's take some questions. What is The PDB primarily store information about. Thank you. Yeah. PDB primarily store the information about three-dimensional structure of proteins and other biomolecules. So it's a structure database. So which section of PDB file contains a brief description of the structure, whether it's a connect section, atom section, or header section. So given we have a PDB file format, so which section provides a brief description about the structure? Yeah, it is the header section. Header section of a PDB file contains a brief description of the structure. So what information does the atom section of a PDB typically contain? Now that we know header section has a brief description about the protein, it's the atomic coordinates of each atom. So we have one more section, that is a connect section of a PDB. So what does it describe? Thank you for your answers. I think okay. Okay, now let's see how a PDB database looks and how to visualize and extract data using a PDB database. So in a Google search bar, one can type PDB, enter. Then you can see RCSP PDB homepage. So this is the homepage of PDB database. So which includes deposits, search, visualize, analyze, download, learn about, documentation and other things. So in the top center, you can see the search bar. So which shows enter search terms, entry IDs or sequences. Okay, let me for example take any PDB ID 1C3W. So this is the results page for 1C3W. So the first tab stands for structure summary, then followed by structure, annotations, experiments, sequence, genome, and versions. Even in the header section of PDB file, when we observe, the experimental details has been given. So when we cleanly observe on the left hand side, the structure has been displayed. And when we closely observe this top section of results, experimental data snapshot. So this structure is solved by X-ray diffraction method. Similarly, the experimental literature regarding this will be provided in the bottom. So when we move into the structure tab, so this is the structure of the protein having PDB ID 1C3W. So several other structure based tools have been given to measure, to search for the structure motifs, to analyze the components present, to analyze the density, to visualize the density of protein and also perform the quality measurement. So this is visualization part. How to download a PDB file? So a PDB file, so we have here display files option and download files option. So we can select download files option and we can select PDB format. So your file will be downloaded. Okay, this is how a PDB database looks to access the data. So what happens? We can download the file, but how to read? It's like reading just the header section, reading just the atom section, or reading just the connect section doesn't make us understand better about the protein structure and its three-dimensional arrangements. So there are many protein visualizers. So the first one is PyMol, which is a powerful molecular visualization tool that is widely used in field of structural biology. So it allows users to visualize and analyze protein structures in 3D and can generate high quality images and videos. PyMol is highly customizable with range of plugins and scripts available to extend its functionality. So the more use of PyMol will be dealt in the later sections of this course where we understand the molecular docking and other concepts. Then we have Chimera. So Chimera is designed for interactive exploration of large molecular structures. It includes a wide range of features, including support for virtual reality headsets. Then we have VMD, so Visual Molecular Dynamics. So VMD is also one of the main software that is used in analyzing the trajectories and in visualizing the trajectory files of MD simulation or GROMAC studies, molecular dynamics studies. That's what. It includes advanced features such as supporting multiple graphic cards, interactive molecular dynamic simulations and scripting capabilities. Followed by that, we have some casual visualizers such as UCSF Chimera, CN3D, and JMO. So, when we closely visualize proteins, there are diverse class of biological molecules. So, despite their diversity, proteins can be broadly classified into some structural classes based on their overall fold and topology. So, the first is alpha helix proteins, that is alpha proteins in short to call. These proteins are predominantly alpha helical secondary structures where the peptide backbone is tightly coiled into helix. Then we have beta proteins or beta sheet proteins. These proteins have predominantly beta sheet secondary structure where the peptide backbone forms extended beta strands. Then we have alpha beta, so which have both alpha helical and beta sheet secondary structures. So the alpha helices and beta sheets are arranged in distinct regions in the protein, forming a domain kind of a structure. So even there are structural classification systems available to classify these. So when we take each class of proteins, each has its own distinct characteristics and functions, and understanding these structural classes is essential for understanding the structure and function of the proteins in living organisms. So SCOP, SCOP and CAT-CH are two widely used protein structure classification systems that provide a hierarchical organization of protein structures based on their evolutionary and structural relationships. So starting with the SCOP, SCOP is a database that classifies proteins based on their structural and evolutionary relationships. So the classification is hierarchical and is organized into four levels, that is class, fold, superfamily and family. So here, sorry, class. So proteins are initially classified into one of the seven broad classes based on their overall shape and architecture. So within each class, proteins are further classified into folds based on their secondary structure content in the arrangement. Folds are grouped into superfamilies based on their structural and functional similarity. And finally, each superfamily is further subdivided into families based on their sequence and functional similarity. For example, the protein lactate dehydrogenase, which has PDB ID of 1LDN, is classified as class alpha beta protein fold. Roseman like alpha beta protein or alpha sandwich fold. Superfamilies lactate or malate dehydrogenase, and families lactate dehydrogenase like. Similarly, we have second system, that is CAT, which stands for class, architecture, topology and homologous superfamily, which uses combination of structure based and sequence based information to classify proteins. So for example, similarly, when we take the representation of lactate dehydrogenase, that is PDB-ID of 1LDN, so the class is alpha beta protein, architecture is Rossmann fold, topology is NAD binding Rossmann fold domains, and homologous superfamily is lactate malate dehydrogenase like. So both SCOP and CAT provide the organization of protein structures based on their evolutionary and structural relationship. SCOP uses combination of sequence and structural similarity by sequence, and the CAT uses a combination of structure based and sequence based information. So both systems are widely used, and these are uh, databases that are regularly updated and have advanced information on these classification of protein structures. So what are the two widely used protein structure classification systems? Yeah, so SCOP and CAT as we just discussed. So how many levels of classification does SCOP utilize? It uses four classification system, that is four hierarchical levels: class, fold, superfamily and family. So we have studied an example. So according to what type of fold does the protein lactate dehydrogenase with PDB-ID 1LDN belong to? So it is Rosman-like fold. So before moving on to the next session, in last class we have discussed about CD hit, and I've shown we have solved a manual example of classifying or building a non-redundant set from a redundant set. So we'll see in a real-time example. So I have generated a Python code which uses basic clustering concepts to group the proteins. So here first I defined a function to calculate the distance between two atoms. So this two atoms means distance between two atoms in two different sequences. So this will final output of this function will be a pairwise distance matrix. Then so first I defined a function to calculate the identity of two sequences, that is pairwise alignment, and to get the identity score. Then I was using a CD hit clustering, so basic clustering I'm doing it here. So the output of this function will be a list of clusters where each cluster is a list of sequence record itself. So here I have given a set of sequences, some of them are similar to each other and some of them are not. So similarly, I have given 20 sequences as input and I have defined the identity threshold of 0.8 and I perform clustering. So when I run this cell, let's see what will be the output. So when we see the output, the first is cluster 1. So under the cluster 1, we see sequence 1, 2, 3. 3 sequences under the cluster 1. And then we see cluster 2. And we see sequence 1 to 8. Then we see cluster 3, which has 8 sequences under it again. And finally, we see cluster 4 with only single sequence. So this means that this single sequence doesn't share the identity of more than 0.8 with any of these sequences falling in cluster 1, 2, 3. So it has been made into a individual cluster itself. So here the examples are very identical, just to make simple for your visual representation. In real time, the sequences will be diverse, so accordingly, the identity threshold can be modified. Okay, I'll send you this file also. You can try with different inputs, modify the inputs that have given. Then continuing with the course is next we'll start with some other types of representations. So the first thing is contact maps, which are graphical representation of pairwise interactions between amino acids and protein structure. So these are commonly used in protein structure prediction and analysis. So the construction of contact map involves two main steps. Determination of pairwise distances between. The first step is to calculate the pairwise distance between all pairs of amino acids in the protein structure. This can be done by using various methods such as Euclidean distance or Manhattan based methods. The second step is to visualize the binary matrix as a contact map. So this can be done by plotting the binary matrix as a heat map. So in addition to heat map representation, today I will be showing other visualization techniques to visualize the other tools. So there are already developed softwares for constructing contact maps such as Contact Map Visualizer, Contact Map Explorer and Contact Map Analyzer. So these tools provide various options for customizing the visualization of the contact map, and which include customization include adjusting the threshold for binary values, selecting the color scheme and adding labels. So with the advancements that are available in coding nowadays, so contact maps has been easy to generate. So to develop a contact map, so firstly I'm calculating the distance between two atoms. Then I'll be calling a function to generate a contact map. So if the distance between two atoms is less than the cutoff, count it as a contact. If it is beyond the cutoff, count it as not a contact. So for examples of all this, I'm considering the same PDB file 1C3W, which is very well annotated in PDB database. So your results you can compare with the PDB database as well. So for here, I'm only selecting chain A for the simplicity of the plot. If you select all the chains, the plot looks clumsy. And I'm selecting a distance cutoff of 5.5 angstroms, assuming that this cutoff will include all the possible interactions, cover all the possible. So the cutoff can be increased according to the need. And the final contact map can be seen like this, with residue index falling on the both sides. So here this heat map shows the closeness of two residues. So this is a demonstration for a contact map, how to develop a contact map. So this based on the analysis part. So this is atom-atom contact map that have generated. So residue can also be generated considering only the C alpha atoms, or there are several conditions defined to make a residue contact maps. So moving next, we have some structural feature related to. So the first thing is interresidue contacts in proteins, which refers to the spatial relationships between amino acid residues in the protein structure. So these interresidue contacts play a crucial role in determining the folding and stability of proteins, as well as their interactions with the other molecules. The first is hydrogen bonds, which are formed between hydrogen atom of one amino acid and the electronegative atom of another amino acid. So these hydrogen bonds are termed to be very important for stabilizing the secondary structures. Then we have salt bridges, which are electrostatic interactions between oppositely charged amino acids. Salt bridges play a critical role in protein-protein interactions. Then we have van der Waals, which are weak and strong range interaction between non-polar amino acids. So these van der Waals interactions play important role in stabilizing the tertiary structures. Van der Waals interactions helps to stabilize the packing of amino acids in the core. Then we have other things such as disulfide bonds, aromatic interactions, cation aromatic interactions, sulfur aromatic interactions. So the identification of interresidue contacts in proteins can be done using experimental techniques. But however, these techniques can be time consuming and difficult, particularly for large or flexible proteins. Therefore, computational methods have been developed to predict the interresidue contacts from protein sequences or structures based on statistical or machine learning models. So these predictions can be useful for understanding the protein folding. So similarly, we look into short range, medium range and long range contacts also. Short range contacts are typically defined as the contacts between amino acid residues that are close in sequence, usually within a few amino acids of each other, whereas medium range contacts are between residues that are far apart in sequence, usually separated by 4 to 10 amino acids we take. And the number of amino acids that we consider may change based on the protein or the class or the type of the protein we analyze. Finally, we have long range contacts, so which are separated by more amino acids. So let's see how these can be calculated. So for this demonstration, I am using one of the tool that is developed by our lab. So I'm using a PDB param. So that is PDB parameter server. So it is available under www.iitm.ac.in by info link. So the name is PDB param, which is an online resource for structural parameters of proteins. So here I'll be selecting compute option. So I'll be entering a PDB ID 1C3W, which we used for visualization as well. Then I'll click on interresidue interactions and submit. So here we have short range interactions, medium range interactions and long range interactions. There are some other protein related parameters which are used in several studies of protein, particularly machine learning models such as contact order, long range order, number of contacts are also present. So I'm just submit. Yeah. So list of short range contacts that are present have been given, followed by list of medium range contacts. then followed by list of long range contacts. Similarly, to visualize what type of non-covalent interactions are present, number of non-covalent interactions present in your protein. So I'm selecting the physiochemical properties, then click submit. So here I can see main chain hydrogen bonds, main chain hydrogen bonds, side chain hydrogen bonds, hydrophobic interactions. So here we have got list of hydrophobic interactions in the protein 1C3W followed by main chain hydrogen bonds parameter. So here the server is unable to extract main chain hydrogen bonds because of the license expiry from HB plus. So one can use HB plus to understand about the hydrogen bonds in a protein. That is currently one of the best tool that is available to analyze the hydrogen bonds present in a protein. So to know more about the PDB param, so under features, several parameters have been provided as hyperlinks, where once you click this hyperlinks, the explanation of each is provided. The explanation of each is provided as a PDF file. Similarly, we have a tutorial developed which makes easy to access the PDB param server. Then we have solvent accessible surface area calculation. So this is the surface area that is exposed to the portion of the protein surface that is accessible to the solvent molecules. So this is important for understanding protein-protein interactions, ligand binding and protein stability. So the calculation of ASA, that is solvent accessible surface area, is typically based on solvent excluded surface model, which assumes the protein surface is formed by boundaries between the proteins and surrounding water molecules. So here we have an algorithm, that is a rolling probe algorithm, which involves placing a probe sphere of given radius. For example, the general probe radius is 1.4. I'm showing this is the van der Waals distance between O and H atoms that have been bonded in the. So 1.4 is the probe or sphere radius. So then we roll the sphere along the surface to identify the points where sphere first touches the protein. So the points where the probe sphere touches the protein surface is considered to be the surface atoms, and the remaining points are considered to be the interior atoms. So then the ASA is calculated by summing up the surface areas of all the surface atoms, and the surface area of each atom is determined by the area of the sphere that is not covered by the protein. So these areas can be calculated using geometric formulas. So there are several methods available, such as N-Access, DSSP, NetSurfP, RVPNet, and Scratch. Currently, we are having one Python module that is called as FreeSASA module, which is fast and reliable method for calculating the solvent accessible surface area of proteins, and it also provides us with a modification of probe radius. Okay, in case of globular proteins, we are using water as a probe radius. Water as a probe. So the radius is 1.4 Armstrong. What if we want to study membrane proteins? We cannot use water, right? So the probe must be changed. So the most repeating part of the membrane structure repeated by it is CH2. So CH2 probe radius is 2. So the solvent accessibility calculation when CH2 is as environment or surrounding solvent can be performed using FreeSASA module of Python. They had two terms that are specifically developed, that is long range order and contact order. So contact order and long range order are two terms that defines importance of local and non-local contacts in the protein. So this tells how important the contacts between the residues which are far in sequence and close in space is important for proteins. So this thing we'll understand. I think that is clear based on the question here. So these are the two formulas that have been defined on how contact order and long range order is important. So these contact order and long range order are parameters that define or that gives a numerical value to the importance of non-local contacts. So what I mean a non-local contact here means residues which are far in sequence. For example, I can tell second residue and the 11th residue, or no, second residue and 20th residue, which are separated by 18 residues. But still, when we see the three-dimensional structure of a protein, they might be interacting with each other. So that this tells. So how a contact order is solved, we'll be checking in the assignment. Today, I have only two main questions on assignment for them. So here I have first question for the assignment today. So yeah, this long range order I'll be explaining clearly. So given the solvent accessibilities, someone is unmute. Please mute yourself. So given the solvent accessibilities of residues, so based on the solvent residues which are more accessible to the solvent, so residues which have more solvent accessibility are with value 56 and value 78, so which is glutamine 8 and alanine 11. So given the accessibility of arginine 74 in folded state and extended states are 78 and 165, what is the percentage of accessibility is?
Sir, excuse me.
Yes, please.
Sir, what was the answer of the last question?
Yeah, GLU 8 and ALA 11, because their values are, one is 56 and other is 78. So solvent accessibility is more for surface residues, solvent accessibility is less for buried residues. Okay, can I move forward? So given the accessibility of residues in folded and extended. So extended state, you can consider it as an unfolded state. So what is the percentage of accessibility is? So accessibility is the term that is related to folded state. Yeah, so it is 47.2. So how the percentage of accessibility of a particular residues is calculated by dividing the solvent accessibility of a residue by the maximum possible solvent accessibility for that residue. So in the current system, current time, folded state, the accessibility is 78 Armstrong square, and the maximum possible, so the residue is mostly exposed in unfolded state. So that is 165. So 78 divided by 165 into 100 gives 47.2. Okay, given a protein has 20 alanine residues on surface, so the buriedness of alanine. So this is also one of the feature that PDB param calculates. So here is how the buriedness of a protein is calculated as number of residues of I type, number of residues in the interior of a protein divided by total number of residues I. So for example, I is a type of a protein. So number of alanines in the interior of a protein divided by total number of alanines. So using this formula, we can calculate the buriedness of the protein given. So make a note here, I told 5 residues on surface, but buriedness deals with the interior proteins. So how to get number of residues in the interior? So total alanine residues minus exposed divided by total residues into 100. So it is 15 divided by 20 into 100. So it is 3 by 4 into 100. 75%. Yeah, yes. So buriedness is calculated such a way. The main factor, the buriedness is studied to mainly classify the globular and membrane proteins in one study. So buriedness of particular residues is observed more in one class of proteins, and buriedness of a particular residues are observed more in other class of the proteins. So this parameter is mostly observed in when we are predicting the topology studies, protein class studies, and functional studies. So based on this buried residues, the transfer free energy of a particular protein. So why one need to study a transfer free energy? When we see folding of some particular class of proteins, particularly membrane proteins, they need a transfer free energy because their the point of synthesis and the point of function is different. So this concept transfer free energy has been introduced based on this buriedness. And in recent study, we also proved that this transfer free energy is crucial in predicting the stability value of a membrane protein, particularly alpha helical membrane protein. So when we take alpha helical membrane proteins, the transfer free energy, using transfer free energy values, we can calculate its stability value. Stability is nothing but the folding free energy difference between the folded and unfolded state. Okay, so here I have one question on contact order. So a protein has 50 residues and 200 contacts. So residue alanine 45 has contacts with ASP 46, GLY 47, ALA 44, VL 71, and ILE 60. So what would be the contact order of the residue? So here I am giving the formula for contact order as well. So kindly calculate. Okay, here the contact order first considers the residues that are close in space. So Sij considers the residues that are close in within the particular distance, but it also takes the sequence separation between them. So if they are close in space, what is the sequence separation? So this gives the importance of local and non-local, that is close and long range contacts. This is contact order. So the contact order is calculated by considering the residues that are close in space and then identifying their sequence separation and normalizing them with the number of residues and number of contacts. Okay, let's try solving this. So given we have number of residues, L has 50. So these residues are forming 200 contacts here. Thank you for your answer. That is correct. So when we see the sequence separation here, the first residue sequence separation is 1, followed by the second residue sequence separation is 2, that is 45 and 47. Next is the sequence separation of 40, alanine 45 and 44 residue. Then we have sequence separation of 26 plus 15 divided by 50 into 200. So this gives 0.045. So this is the contact order of this protein.
Sir,
Yes.
maybe you can answer now or later also. What is the meaning of this contact order 0.0045, where we use and how we can utilize that?
That's all. These parameters are useful when people who are computationally studying protein folding. So you can refer to my recent paper TMH STAP rate, where you will see what is the importance of contact order in folding alpha helical transmembrane proteins. TMH STAP rate by Ramakrishna Reddy.
Next week when you are coming, 2 to 3 slides kindly explain what excitement, that will be really exciting that you have done this, and you can describe only 2 to 3 slides.
See, actually contact order and its important to stability will come when we are talking with protein stability class. This is just an introduction to the calculation part of protein
Okay,
features.
thank you.
So similarly we have features for long range order. So here the slight change in long range order is long range order takes only the importance of long range interactions. So the value is assigned, a binary value is assigned only if a protein is having a sequence separation of more than 12. So we'll try to solve this.
Sir, I am sure you know better because you are from the same lab.
All details of this, how it is implemented, the applicability, the applications, all are given in the reference term. So here, when we consider the residues, similar residues here, a binary value of 1 will be assigned only if the residue sequence separation is more than 12. So in most of our cases, the residue separation is less than 1. Only in valine 71 and isoleucine 60, we have sequence separation. So so only in 2 cases we have binary value. In rest all cases it is 0.